Good morning. In this session, we are continuing in connection with the anomalous secondary growth in Monocot stem, that is Dracaena, and the morpho-anatomical, that is internal morphological, in relation to the anatomical features which we can see from the axiomatic sketch and the different parts of the morpho-anatomical differentiation and how the anatomical secondary growth and anomalous nature which is exhibited in this plan, which we can discuss in this session. Okay, coming to the diagrammatic level, here you can see normally or naturally the stem, which consists of primary or airly formed vascular bundles, and also it is with a number of secondary and tertiary or second or third row of vascular bundles which is formed. So we are coming to Dracaena. First of all, the internal level, what we can observe that there are larger bundles are there at the center, and actually these bundles are the primary or early formed bundles during primary thickening, and such bundles are getting disposed or distributed or randomly or irregularly distributed inside the ground tissue of the stem. And there we can see the bundles are conjoined and collateral, and they show the outer phloem and inner xylem and both the vascular elements devoid of the cambium at the center. So that is why the bundles are called closed bundles. Cambium is absent, that is fascicular cambium in between xylem and phloem, that is the cambium is absent. So closed bundles which are formed at the primary thickening. And coming to the secondary thickening, naturally the changes or variation is in some of the monocotyledonous members. We can see the primary bundles become enlarged to some extent, and the addition of secondary cortex, that is parenchyma cells, which enunciate a slight increase in girth and which is assumed to be the secondary thickening in monocotyledonous plants. But coming to here in this particular example, that is in Dracaena, we can see the early formed bundles which is kept as such in the center portion as in the ground tissue, and thereafter there are second or third ring of vascular bundles which are formed. So from where the vascular tissues are formed? Normally the vascular tissues are developed from the cambial initial, that is the vascular cambial cells. So here in these areas in the primary structure, it occupies with the ground parenchyma tissue, and here and there or intermittently the ground parenchyma tissue become differentiated as primary thickening meristem, that is etium, and this primary thickening meristem act as the procambial strands, because here and there the strands or bits of parenchyma cells which are very differentiated into primary thickening meristem, that is the vascular cambial strips. And such vascular cambial strips become multi-layered in nature, and that multi-layered nature which leads to the development of multiple number or more number of vascular bundle-like structures in the outer region to the airway formed or primary vascular bundles. Okay, so here in the structural differentiation, what we can see the outer region. So in connection with such abnormal behavior of multi-layered cambium, which develops the alternating band. So here you can see one bundle and here is another one, and the outer ring is or outer bundle which is formed in between or alternating to the inner bundles, the second row of bundles are formed. So it produces or it seems to be the alternating bands of concentric rings, which is a continuous ring of vascular bundles which is formed. So that is the anomalous nature which is seen in Dracaena. And in addition to that, the outer rings or second row or third row of bundles, which are usually due to the thrust of more development of concentric alternating bands of bundles, which makes the congestion or destruction of the newly formed young bundles, and to devoid of such denaturation or destruction of such new young bundles. To avoid that, those outer cortical cells or ground parenchymatous tissue which become thick-walled in nature, and they are highly lignified conjunctive tissue. So their cells become lignified. So what we can see in the outer ring, the bundles are engulfed in the lignified conjunctive parenchymatous tissue, and the inner layers, which is seems to be the bundles or xylem and phloem, which is engulfed in the ground or cortical parenchymatous tissue. And here the structure we can see in the outer region, it is with the suberized core, it is highly suberized or suberized deposited parenchymatous cells, and as part of the formation of the perineum or due to the development of formation of fourth cambium, that is phalange, which is formed in the outer region, and it is the fourth cambium which derives outer phellum or core and inner phalloderm or secondary cortex. So, and in addition to that, the outer epidermal region become broken due to the formation of the perineum, and here and there we can see there are a number of lenticels which will help for additional aeration to the inner tissues of the core cambium and also the secondary cortex. So lenticels are also present in the outer region. And in some of the species, either it is green or brown. Dracaena, we can see the secondary cortex which possesses the pigmented cells, either it is green or phycocyanin or phycocyanin like colored pigments which are seen in the secondary cortical tissue. And inner to the secondary cortex, what we can see there is the multi-layered cambium which is formed. It is one of the anomalous behavior in Dracaena or in this monocotyledonous plants. And so here we can see the multilayered cambium, lignified conjunctive tissue, and the bundles with the phloem and also xylem. Phloem is completely surrounded with the xylem. That is why the bundles which are formed during secondary growth in thickness in the species, which is due to the activity of abnormal behavior or anomalous behavior of cambium, and which forms the amphivascular bundle. That is, the phloem is completely surrounded with the xylem elements. And these phloem, xylem, and the secondary vascular bundles, which are actually as part of the secondary tissues. That is, it is developed during secondary growth in thickness, and they are considered as secondary tissues. And here we can see third and second and third bundles are there, and they are generally in alternating bands of concentric rings of vascular bundles. So, and coming to the inner portions, here we can see the ground tissue is there, and they are thin-walled parenchymatous ground tissue, and in the center region or somewhat middle to the cross-sectional of stem, we can see it is with the parenchymatous ground tissue, and the phloem and xylem is arranged as said in the early description. The early formed vascular bundle with the outer phloem and inner xylem, and coming to the secondary bundles, the phloem is completely surrounded with the xylem. So the early formed bundles with the outer sphere and bundle shape, and it is a typical monocotyledonous stem character. And the second and third row of bundles which is formed, and those bundles are amphifossal in nature, and they are alternating in bands, and which are developed from the secondary meristem. That is, it is from the primary thickening meristem or derivatives of primary thickening meristem, which is called the vascular I think it is called the vascular bundles, and they are collateral in the early form, and coming to the later ones, they are anti-vascular bundle. And these early formed ones are grouped under, that is the parenchymal ground tissue, phloem, xylem, that is the endoxylem, and collateral vascular bundles, which are togetherly considered as the primary tissues, and the outer secondary and second and third row of bundles which is formed, they are considered as the secondary tissues. So this is the structural nature of Dracaena, what we can see from a cross section or from a diagrammatic description. Okay, so thank you.